Hi, my name is Selena Chen, and this is my third year in the BioScience program, and thank you for coming to our talks today. So when most of us think about bacteria, we think about how they cause diseases, like pneumonia, tuberculosis, leprosy, and many others. However, the truth is that 95% of bacteria don't cause any disease at all. In fact, some bacteria can potentially be used to treat disease, maybe even heart attacks. So let me show you how this might work. So this is a beating heart. When a heart attack happens, an artery that supplies oxygen to the heart muscle becomes obstructed. This means that the blood and oxygen that normally flow through this artery and feed the downstream muscles can no longer get through. Therefore, the heart muscles downstream of the obstruction do not get enough oxygen, and this is called hypoxia. And when hypoxia lasts for more than just a few minutes, that is when heart muscle begins to die. Now, to treat this condition, doctors have already developed several methods of successfully removing the obstruction in the artery and allowing blood to flow back to the heart muscle again. However, until the moment that the obstruction is removed, there is still no oxygen flowing to the heart muscle. In other words, the heart muscle is still dying. So how can this muscle be provided with oxygen during the time that the obstruction is being removed? Is blood the only source of oxygen, or is there another source? A couple of researchers had an interesting idea. How about taking advantage of photosynthesis? So when sun shines on any photosynthetic organism, such as a plant, when carbon dioxide and water are present, then photosynthesis takes place. Then glucose and oxygen are produced. But obviously, we can't grow plants in your heart muscle to provide oxygen. Luckily, there are other photosynthetic organisms, such as photosynthetic bacteria, that do have the ability to produce oxygen. So let me show you how researchers use these bacteria to save the dying heart muscle. A group of researchers at Stanford University, led by Jeffrey Cohen, started by inducing a heart attack in a mouse's heart. They did this by tying off one of the heart arteries. Because of this, the heart muscle downstream of the tie did not receive any blood or oxygen. In other words, the heart muscle was hypoxic. Now, to save the dying heart muscle, the researchers injected live photosynthetic bacteria that were producing oxygen directly into the dying heart muscle. Then after 1 hour, the researchers removed the tie from the artery, allowing blood and oxygen to flow once again to the heart muscle. The results of this experiment were very encouraging. When the researchers compared the heart that had not been injected with photosynthetic bacteria with the heart that had been injected, they found out that after 24 hours of recovery, the amount of heart muscle that was still dying was far less in the heart treated with photosynthetic bacteria. They also found out that the immune system had cleared all the bacteria from the heart. In addition, there was never any sign of infection in the heart or anywhere else in the mouse's body. Lastly, after 4 weeks of recovery, the treated heart was functioning significantly better than the untreated heart. Thus, the researchers concluded that the photosynthetic bacteria had a protective effect on hypoxic muscles, presumably due to the oxygen that the bacteria provided. I found this experiment really intriguing and started to wonder if it might be used to save other essential organs that might become hypoxic. The organ I decided to focus on was the kidney. Each person has 2 kidneys. There are a pair of bean-shaped organs located in the back of your torso on either side of your spine. So why is the kidney that important anyways? Well, the kidney is probably best known for its ability to filter waste and toxins from the bloodstream and excrete them into the urine. But what you might not know is that the kidney also regulates blood pressure, regulates acid-base balance in the blood, and controls our hydration status. And if that's not enough, it also regulates deposition of calcium into the bone and regulates the production of the red blood cells. So now I bet you can understand why the kidney is an essential organ. So that is why I decided to do my project on using photosynthetic bacteria to treat hypoxic kidney damage, a theoretical study. So my hypothesis was the photosynthetic bacteria can be used to prevent or treat hypoxic kidney damage. My methods were very different from most other bioscience projects, and I decided to gather evidence from a wide range of sources and then try to fit them all together to test my hypothesis. So the first question in this process was to address the following question. Does a medical situation ever even exist that would need this type of treatment? If not, then there is no reason to continue this line of investigation. To find the answer, I spoke with an expert on kidney disease, Dr. Thomas Devon, who is a nephrologist that treats patients with kidney disease. I told him about my hypothesis, and he indicated that there is a medical condition in which this treatment would be really helpful. This condition is called acute kidney necrosis, which is the sudden death of a portion of the kidney tissue due to loss of the blood supply. One example of when this might happen would be a serious car accident when the blood vessel supplying the kidney might be damaged. Thus, the supply of blood and oxygen to the kidney is disrupted. Does it sound familiar? This would cause kidney hypoxia. And during the time when the surgeons are repairing the blood vessel, kidney tissue would be dying due to lack of oxygen. So this could be a situation where photosynthetic bacteria could temporarily supply oxygen until blood flow can be restored. So once I found out that there was a medical condition for which this treatment can be used, I needed to determine if photosynthetic bacteria can even survive in the human body. Well, Dr. Cohen's study demonstrated that photosynthetic bacteria can exist and function in a live mouse's heart. Since the physiologic conditions in the mouse body is very similar to the human body, I believe I can say that it is very likely that photosynthetic bacteria can survive in the human body. Next, I needed to find evidence to answer the question, can photosynthetic bacteria survive in the presence of kidney cells? After many hours of searching the scientific literature, I was not able to find any direct evidence that supported this. But I did find canine dogs' kidney cells have been successfully grown in the presence of bacteria, and that these same cells have been successfully grown in the presence of influenza virus. I also found articles showing that many other mammalian cells could be grown in the presence of bacteria, including liver cells and spleen cells. So from this evidence, I believe that photosynthetic bacteria can possibly survive with kidney cells. Lastly, just because treatment with photosynthetic bacteria works in the heart muscle doesn't mean that it will work in the kidney cells. So the next step is to answer the question, what are the differences between the heart and the kidney? And more importantly, how might those differences impact the use of photosynthetic bacteria to treat hypoxia? When we look at a cross-section of the wall of the heart, we can see that it is quite uniform in appearance. In fact, the heart muscle is made up of only 4 cell types, and 95% of which are heart muscle cells. But when we look at a cross-section of the kidney, we can see that the composition is very diverse. Moreover, the kidney is actually composed of 26 different cell types, which makes the cell composition of the heart and the kidney very different. Therefore, when we inject the heart muscle with bacteria, it is primarily going to produce oxygen for the heart muscle. However, when we inject it into the kidney, we don't really know what cells we're going to treat. A more important difference involves the amount of blood vessels in each organ. When we look at the wall of the heart again, we can see that there are very few blood vessels. However, when we look at the kidney, there are blood vessels everywhere. This has 2 implications. First, there's a much higher risk of extensive bleeding if we were to put needles in the kidney versus in the heart muscle. In fact, the use of this treatment might even be discouraged for this reason. Second, if you recall from the Cohen experiment, the bacteria in the heart was cleared in 24 hours. However, in the kidney, where there are many more blood vessels, it is possible that bacteria will be cleared away before it can provide enough oxygen. So, given that the kidney has so many different cell types and is full of blood vessels, I would say that the heart and the kidney will probably not respond similarly to the treatment. Thus, in conclusion, the evidence that I gathered does not support my hypothesis that photosynthetic bacteria can be used to treat hypoxic kidney damage. However, this evidence does seem to indicate that further studies in this line of research will be warranted. Thank you.